great to be here. So I'm so excited today to present here at our Kids First Spring webinar, showing about variant discovery and Kids First data sets to share a little bit of an additional deeper dive as well. But before we do that, I do want to acknowledge that behind us is a team of dozens of people who work very, very hard to make all of this possible. So just want to acknowledge all of us at the Kids First Data Resource Center. Everybody has put a lot of effort into this presentation, what we're about to share today, and I wanted to acknowledge them and thank them for all their hard work that's gone into this. All right. So if you haven't been to explore the Kids First website recently, the first thing I wanted to share is that we have not just a new portal, but also a new website. And so if you navigate to kidsfirstdrc.org, you'll see a completely updated page with new content. And it really does a great job of highlighting what we are, what we do, and the value of what we've built. So after the webinar today, if you haven't seen it, please check out our website, kidsfirstdrc.org, and read more about us. Of course, and additionally supported with that is that we have a whole new help center. And so attached to the website is guides and with pictures and screenshots and short little videos about how to use our portals and platforms if you ever get stuck and need some further help. So today we're announcing that we're launched a new version of the Kids First portal. And so the portal is available at portal.kidsfirstdrc.org. Highlighting that we have our previous version of our portal will soon be going away here coming up in the middle of June. We're going to be depreciating that older portal and we'll only have the new portal. So some of the new features you'll notice right on the homepage is that we now have an updated kind of underneath the layer of the portals that we have updated servers. So now the Kids First data is stored in what's called a Fire server, which allows for API authorization for advanced users. We've also enabled connections with the Kavatica Variant Workbench, which we're going to hear a little bit more about from Jared in the second half of the demo today. Up at the top, we've improved the studies page. So now the studies page includes additional detailed information about the different data types and experimental strategies of the different data within each study. And the data exploration tool has been improved because we've combined the data exploration and file repo pages into one single page. And so now you can search for participant file specimens and data files all on one page, which I'll show here in my demo in a couple minutes. We've also made improvements to our variants page as well. And so the variants page allows you to be able to search and explore variants in the Kids First data sets and gives you summary level variant exploration. So we'll show a little bit of an example of how to use the variant search tool in our demonstration. And so there's going to be two parts today. So my part is part one, where we're going to be exploring the data on the Kids First portal. So we're going to try to identify participants, look at genomic variants, and then figure out how to review studies and apply for access. And then after that, I'm going to pass some data to our cloud platform, Kavatica, and I'm also going to pass the baton to Jared, and he's going to show us how to use Kavatica's Variant Workbench to identify genomic variants within those participants, and then pair the variants with annotations as well. So a demo, I think, is even better when we have a specific example, and we're so fortunate that Dr. Rao gave that great background about orofacial clefts. So I don't need to go into too much detail because I am a little short on time, but orofacial clefts are caused by the failure of tissue to fuse properly during fetal development. They're a relatively common birth defect, can cause as a result of both genetic and environmental causes, and multiple developmental pathways have been implicated in orofacial clefts. And so as Dr. Rao shared in her intro, she recently was part of a group that published a paper identifying the gene AMOTL1 and variants in it associated with not just orofacial clefting, but also cardiac anomalies and tall stature. So her and her research partners used the orofacial cleft cohort data sets as a secondary cohort to kind of validate their findings. And so again, to highlight the example here, so they were able to identify this hotspot here in positions amino acid 157 through 161. And so at this time, and so to pull out a specific quote from the article, Dr. Rao and the authors identified that identifying the individuals that have these variants were difficult at the time. And so as a result, they needed to download the trios in order to be able to find participants that had that variant. So what we're going to show today is that you can now do that on the Kavatica platform and the Kids First portal to get started. So we've improved the process for users in the future. And so the demo that I'm going to share today, if we have a more generic kind of view of it, is a lot of the questions that we get are what types of participants in Kids First data sets are diagnosed with my condition of interest? Do participants in Kids First have variants in my gene of interest? And how do I apply for access to the genomic data for these participants? And so we're going to answer that by showing 3 different pages. So we're going to answer the first one by looking at the newly designed data exploration page. We're going to look at variants in the genes of interest using the improved variant exploration tool. And then we're going to explore the genomic data and how to apply for those by looking at the updated studies page. And then again, we're going to use our example. So today our condition of interest that we're going to look into is orifacial clefts, and our gene of interest is going to be AMLTL1. So at this point, I'm going to navigate to our website. And so anyone can do this following the presentation today. Navigate to portal.kidsfirstdrc.org. And when you navigate to the dashboard, so you notice that everything, it has a different look. We have a new sleeker dashboard with some similar kind of cards or widgets that explain different aspects as far as your authorized data and your connected Kavatica projects. But I'm going to start with the data exploration. And so our first question was, we want to identify which participants in Kids First are diagnosed with my condition of interest. And so we can do that. If we scroll down, originally it gives you a big overview of different data modalities and data types and information about phenotypes. But we can search specifically for, if we go into our clinical tab and apply a clinical filter for our HPO term for oral clefts. So we use on our portal, we use the HPO, human phenotype ontology. And so if I type in oral cleft, then it pulls up oral cleft, which is kind of a parent term that includes both cleft lip and cleft palate underneath it. So then I can apply that term. And so it tells me that we were able to identify 1,517 participants who are labeled with that condition. If we scroll over to the participants tab, so now without actually changing the page, we can just switch between these different tabs. We can learn more about these different individuals. And so most of the individuals with the orofacial cleft phenotypes are in our Kids First orofacial cleft studies. So like the Latin American Family Study or the European Ancestry Study. But you'll also notice that there are additional studies in here as well. So for example, this is the Kids First intersection of childhood cancer and birth defects. So supporting that, the Kids First data set is well annotated across multiple studies for our different phenotype conditions. And so you can use these additional filters to explore what different studies are available or what studies have participants with these particular conditions. So if we look at our study name, again, the most frequent or largest studies with the largest number of participants, again, are the orofacial cleft studies. But we do have participants in the smaller number of participants in our other studies, like our chromosome 18 copy number study or charge syndrome study. So they're to kind of connect the conditions across the multiple different Kids First studies. From there, you can then click across to learn more about the biospecimens associated with those participants, or into our data files where you can then access the individual alignments and variants that are associated with these participants. So I'm going to skip ahead to our variants tab here. And so this lets you search for particular variants of interest. And so, for example, if we wanted to search for a gene, for example, we could type in the name of our favorite gene. And so we might try to look into that study that Dr. Rao shared, and maybe we're interested in AMOTL1. And so we can apply that filter, and then it pulls up a list of variants that are present in Kids First data sets in the AMOTL1 study. at this point, and it's sorted by deleterious consequences. And so many of these are, the most deleterious at the top, and then the missense ones, and then silent ones at the bottom. But we can filter this further. So we can add, for example, an additional filter looking at pathogenicity, looking at CAD score. So CAD score is a filter that predicts how deleterious a particular mutation is. And so if we apply a filter for where your CAD score is greater than 15, and then we want to combine those. So we're looking for variants in AMLTL1 where the CAD score is greater than 15. We get a total of 125 variants. At this point, we can kind of scroll through, and so they're sorted by position in the genome. And so you can see we can actually, if we keep going further and further, we can find that variants in that particular region at the 157th amino acid. So we were able to find the specific variant that Dr. Rao's paper identified, and we're able to learn that there are 2 participants in one kid's first study that have this variant. So we can click on this link and learn a little more about this particular variant. It tells us more information, more annotation information. It tells us about the frequency and explains that in the Kids First European Ancestry Study, there are 2 participants with this particular variant at this site. So if we want to learn more about those participants, though, which 2 participants are they? This is kind of the extent of where we can go on the Kids First portal, because at this point you would need to apply for access to be able to identify who those participants are. So you could do that by going to the studies page, which has a list of all of our Kids First studies. And so if you search for that code, that orofacial cleft of European ancestry, then it pulls up our study here along with the dbGaP ID. So of the 1,400 participants, only 2 of them have this particular variant. And then you could apply for that access by clicking this link, which takes you out to dbGaP. And then you can submit an application to request access for this particular data set. So once you have access, you'll be able to access all of the genome files for this study in Data Exploration tab. But you can also will be able to explore them within the Cavanica Variant Workbench. And so this will allow you to be able to search very rapidly and add these annotations quickly to be able to understand, identify who those participants are.